attitudes concerning topics such as abortion, immigration, the death penalty, and pacifism. And then in the same article, it quotes from John Hibbing, who's a political scientist at the University of Nebraska-Lincoln. And he's quoted as saying, "It is difficult to change someone's mind about political issues because their reactions are rooted in their physiology," close quote. So we note here this sort of assumption of determinism. Genes and physiology are seen as something different from us and from our mind, and they seem to be controlling us, so we can't even change our mind. So I want to suggest a few ways today in which we can think about behavior and genes more accurately from a science perspective, and then go on to think a little bit about how this genetic perspective on humankind opens up, I think, a very natural conversation with Christian theology. But I want to spend a bit of time first on the science. And here, obviously, because of time, we're just going to have time for a very brief introduction to some of the information and ideas that you'll see at greater length in this book, Are We Slaves to Our Genes? It came out a few months ago. And by the way, as I give a plug for the book, let me just mention, if you want, I think the most heavily discounted copy, you can get it from the online shop at the Faraday Institute here in Cambridge. You can shop around, but here you can get it for around $19. Right, a bit of science. Well, the first key to understanding how genetic information is built into our personal human identities is, of course, provided by developmental biology, which is Jeff Hardin's particular field, so I have to be careful what I say here. But let's start with a newly fertilized human egg, a zygote. What's inherited from the parents is not naked DNA, which by itself can do nothing, but a complex system of DNA and RNA and proteins and nutrients that together operate to regulate cell growth and division. And in fact, if you look in the human egg just prior to fertilization, you'll see it contains at least 3,000 proteins. It contains 7,500 different messenger RNAs, thousands of other small molecules which communicate instructions to the new genome as to how to behave and so forth. Coming back to developmental biology, we see that there are two types of environment, if you like. You've got the microenvironment, you've got that environment immediately around the cell and the DNA itself, how DNA is controlled. And then you've got the macro environment. You've got the fallopian tube down which the zygote soon starts traveling, plus the mother herself, plus the mother's own broader environment. And so the DNA and its regulation and these two types of environment are completely integrated right from the moment of fertilization. Key part of the microenvironment of the gene is, of course, other genes. The genome operates as an integrated system. There's no such thing as a selfish gene. There are only genes which are cooperators, and like people, they function according to the company they keep. And in the early zygote, it's not the DNA which causes development to begin, but rather the proteins inside the mother's egg, inherited there from the mother, which regulate which genes in the DNA are switched on and off. I always like to think of proteins as like the players in the DNA orchestra, if you like, that cause the genes to play an integrated symphony of life. And there you've got causal networks also operating in all directions. Now, we obviously don't have time to do full justice to human development biology in the space of a few minutes, but I think it's worth highlighting that the infant brain is not a sort of miniature version of the adult brain, but rather it's a self-organizing system that only self-assembles correctly if the right inputs are available at the right time during early postnatal life. Here in this slide, we see this great increase in synaptic density during the first 24 months. And that occurs in response to the correct environmental inputs, which must happen at the right moment. And then once adulthood is achieved, each neuron receives on average around 5,000 synaptic connections. These are the connections between the different neurons, the different brain cells, although actually the range is very broad. Some neurons actually connect up to other neurons by as many as 200,000 synapses. And since there are around 100 billion neurons in the adult brain, this implies that the brain contains a staggering 500 trillion synapses, 5 times 10 to the 14 or thereabout, plus or minus a few. And so that makes the adult human brain the most complex entity that we know about in the known universe. And it's this synaptic architecture which is constructed during the first few years of human postnatal development. So continuing on then into adulthood until you get to me right there at the top of the screen. Okay, there I am. You get this integrated interactionism between the genes, the microenvironments, the macroenvironments, becoming, I think, even more striking. Because that growing individual makes key lifestyle choices about their diet, about their exercise, levels of stress, alcohol intake or not, smoking or not, clean environment, polluted environment, drug taking or hopefully not, choice of friends, lonely environments, and on we could go. All these factors, myriads of factors, many more, feedback to affect what we call the epigenome, that is to say, those epigenetic modifications of the individual
Hi, sweetheart. So
that regulate the expression profile of their genes. Right, so the second major field that we need to think about when considering whether we're slaves to our genes is that of human behavioral genetics. And this field aims to discover how much of the variation in behavior that exists between people may be explained by genetic variation. And again, this is a vast field that we only have time just to touch on, but just to give you an idea, a flavor. So one aim of behavioral genetics is to discover what is called the heritability of a certain trait. The word trait in this case refers to any human behavioral characteristic that can be measured. And so the heritability of the trait is defined as the proportion of the variance in a trait in a specified population that can be ascribed to genetic variation. Now just look at that very carefully for a moment. It's a statistical estimate, and the definition is rather important. For example, you can present a heritability as a percentage, and that's often misinterpreted as if you're referring to the proportion of inheritance of a trait, when all it really represents is the percentage of the total variance in a particular population. So when we say that the heritability of IQ, let's say, variance is 50%, the unfortunate impression is often given that a particular trait in a particular individual is actually caused 50% by their genes and 50% by their environment. Well, that's not what heritability is about. Heritability is about population variance, and it's a population statistic. Right, so along with behavioral genetics, then we get this other vast field of molecular behavioral genetics. The experimental approach that's received most recent attention is known as Genome-Wide Association Studies, or GWAS. And this approach capitalizes on the millions of what we call single nucleotide polymorphisms, or SNPs, that are present in the human genome. What are SNPs? These are single letter differences in our DNA that act like signposted scattered right across the genome. And it's now routine to genotype people for their SNP profile. So if you like, the SNPs act as sort of flags to mark different segments of the DNA of the genome, which varies slightly between individuals. If a particular SNP keeps on associating with a particular medical or behavioral trait at levels above chance, then the inference is made that there should be nearby in the genome a variant protein encoding gene or gene regulatory region that contributes somehow to the development of the trait in question. And GWAS has been incredibly successful in highlighting the role of genetic variation involved in medical genetics, including psychiatric genetics. But in terms of the kind of variation that exists in the general population with reference to traits like variation in IQ or educational attainment, that kind of thing, I think in a sense, it's been its own gravedigger. And I want to just explain why I make that rather bold assertion. Let's take that example of educational attainment so you can see what I mean. So the heritability of educational attainment is around 40 to 60%. It depends how you measure it. You can measure it by simply, have you been to university or not? How do you do in exams? And there are various ways of of measuring that? Well, this is just one example of a number of recent studies, but in this study, they looked at 1.1 million people of European ancestry, and they identified 1,271 SNP, single nucleotide polymorphisms, that associate with educational attainment. And they do this in a statistically significant way. And if you total them up together, they explain about 11% of the variation in educational attainment in this particular population of European ancestry. And so what this means really, if you average it out, then each of those genetic variants on average, it's an average thing, but might contribute something like 0.02% to the overall variation in that population. In other words, not much. But think about it, that's exactly the kind of result that you would expect from our developmental biology. If you think about the brain, if you think about the thousands of genetic variants that are involved in the early embryological development and postnatal development of our brains, then each one is liable to make a very tiny, tiny difference in the functionality of those adult brains in later life. So then, are we slaves to our genes? Well, we notice, first of all, that there is no gene for any particular behavior. There is no gene for educational attainment. Rather, there are hundreds of thousands of different genetic variants that contribute to the variation that we see in a given population with regard to a particular trait. Our own particular identities and unique personalities, I like to say, are 100% genetic and 100% environmental, because they're all integrating and wedding together all this wonderful complexity. And I like this verse from the Psalmist 139, "For you created my inmost being, you knit me together in my mother's womb." We know that the Psalmist didn't know much about developmental biology, but at the same time, I think knitting is a wonderful picture for this great enterprise of developmental biology, of integrating all these various factors together to produce the person that I am, the person that each one of us may be. And the second thing we notice is that although a single gene is not causal for any particular behavioral trait, there's no such thing, as we say, for educational, no such thing as a gene for educational attainment, we can see that specific gene variants cause differences in a trait, Now, of course, there's a vast number of factors that cause differences in some behavioral trait. We've got our own parental upbringing, we've got our peer group pressure, we've got media, we've got what sport we play, we've got what we eat or maybe what we don't eat, and so forth. Onwards we could go. And the key point to recognize here is that all these factors are integrated together, they're woven together to generate free agents, that we are able to have moral responsibility for the choices that we make. So I think we need to distinguish carefully here between traits that happen to us with traits that we make happen, thereby perhaps becoming habits in the process. But nevertheless, we chose them to be so. Traits that happen to us include items such as personality traits. As childhood development proceeds, very clear personality traits emerge, not out of the child's choice, but as a consequence of developmental biology. These traits are not fixed, can be modified to a greater or less extent in adulthood, but they were not chosen by us as children. And neither are such traits genetically determined, given that variant genes represent but one set of factors amongst many during this developmental process. Other behavioral traits that vary in healthy human populations, put there on the right-hand side of the screen, are things that we make happen. Things like, I don't know, political activism, or going to college, or playing basketball, or hopefully not criminality, whatever it might be. These are things that we choose to do. They don't happen to us. But genetic variants, even in those kinds of things, are still difference makers in that they can lead to the development of people with predispositions to choose in certain ways. If that were not the case, then one would expect the heritability values of all those traits there on the right-hand side of your screen in populations to be zero. And they're not. Each one of those things has a positive heritability. Let's give an example of that. Tall individuals are more likely to choose to play basketball, although there are plenty of short people who choose to play as well. The decision to go to university will, of course, be influenced by one's academic record. That certainly has something to do with genetic variation. Different genomes tend to correlate with different life outcomes in a probabilistic kind of way. But I think it's clear there's nothing in genetics that supports genetic determinism outside of severe medical pathologies, which are not really our topic for today. So contemporary genetics simply does not support the idea that we are slaves to our genes, but we can certainly accept the idea that genes are influencers. Well, you might be thinking by now, this all seems very distant from Christian theology, and I want to say, not a bit of it. I think there is a very fruitful conversation that we can have here between genetics and theology on this topic. I want to focus here particularly on the ancient biblical doctrine of humankind made in the image of God, an idea often called the Imago Dei, just to use the Latin description of that doctrine. And I'd like to suggest that this idea provides a useful bridge between the scientific and theological understandings of personhood. We first find the idea, of course, right back at the beginning of the Bible in Genesis chapter 1, where we read that God created Adam, or humankind, in his own image. In the image of God, he created him, male and female, he created them. Well, clearly, this Adam in this context, as we got on the screen there, is referring to humankind. The whole of humankind are made in the image of God, according to this amazing theological essay we have here in Genesis chapter 1. So, of course, that raises the question, doesn't it, in our minds, well, how would the first readers of Genesis 1 have actually understood that little phrase, image of God? An important clue, I think, comes from the many contemporary texts in the ancient Near East that talk about the king and sometimes priests as being in the divine image of one of the polytheistic gods that were revered by the nations that surrounded Israel. So most likely the early readers of Genesis would have first thought of kingship as they encountered the idea of humankind made in the image of God. Here is a totally new idea about the value and status of humankind. Humankind created with dignity and worth, called to play a delegated kingly role, male and female alike, in caring for the created order. And so the kingly and priestly roles previously allocated to the privileged few by a pantheon of gods were now being delegated instead by the one creator God to the whole of humanity. It's often said that the Imago Dei was being democratized. It was now humankind as a whole who were to play the kingly role. And as one true creator God now cares for the needs of humankind. So it's an original ancient Near East context. Genesis 1 was really what we might say as social and political dynamite. And ever since then, the idea of humankind made in God's image has been used as a very powerful justification for human rights. Now, there are two particular strands in this idea of humankind made in the image of God that I like to bring into conversation with genetics. And the first is the value and status of each human individual, irrespective of their genetic status. I think there was a time when the idea of the image of God was perceived much more as a list of characteristics of humans, such as rationality, creativity, that sort of thing. Those things that distinguish us, humankind, from the rest of the animal kingdom. I think that list is really important, but I don't think it really gets us to the heart of the idea as we see it in Genesis. Instead, we see more the idea of a delegated value and status bestowed by God that exists irrespective of the frailty and transience of human life. Humankind's value lies not in some list of intrinsic qualities, important as they are, but in God's grace, in the bestowal of a kingly, priestly status that we certainly do not deserve. And that status is freely given to the whole of humankind as a community. And those whose genetic endowment entails that they suffer some handicap in life, be it physical or mental or both, are as much sharers of the image of God as anyone else. Where the individual is unable to express or fully practice their kingly priestly functionality, then Christian compassion insists that we care for and protect those less fortunate than ourselves. So the care receiver is as much reflecting their status as being made in the image of God as the caregiver. And I think it's important, isn't it, to highlight this fact. The fact of humankind being made in God's image subverts, it undermines any kind of move to make distinctions between people based on their genetic endowment, or their background, or their culture, or the color of their skin, or anything else about them. All of humankind are made in the image of God. And that is the revolutionary teaching of Genesis chapter 1, and indeed the whole of the rest of the Bible. Now, the second resonance that we note between the idea of the image of God and our genetic identity is its insistence that involves diversity, male and female. He created them. So the image is not some static sort of status quo, a list of fixed human characteristics so much, but a dynamic, ongoing developmental process carried out in relationship, which God delegates ongoing developmental process through the social human community in which male and female equally collaborate. And so being made in the image of God refers to humanity in diversity and in community, not humans as it were, sort of atomized individuals. And this theological framework, I think, coheres very well with the biological story that I've been presenting today, a story that focuses on the development of diversity and that guarantees the uniqueness of every human being who has ever lived or whoever will live on this planet. The genetic diversity that exists between individuals is far greater than we had imagined even a few years ago. And far from being a threat, I just think we need to celebrate that diversity. And we celebrate that human value and dignity that the Imago Dei provides for us. So in closing, what I've been suggesting today is that there is nothing in contemporary genetics, barring some rare but tragic cases of medical genetic pathologies, that in any way subverts personal moral responsibility. Definitely not slaves to our genes, that we may be predisposed to behave in certain ways due to our genetic environment. And we cannot therefore escape our responsibilities to God and to each other, and indeed in terms of our care for the earth around us. We will all be called to account because we have that moral responsibility before God. And so with that sobering thought, I will close. It's over to you for questions and discussion. And I just want to flag up again, sorry for plugging the book several times, but it just gives a much fuller account of what I've been able to introduce today for you so briefly. Thank you very much for your attention.
Well, thank you very much, Denis, for such an excellent presentation. And I know that that whole book being condensed into 30 minutes is very hard to do. So you did an excellent job. First of all, I'd like to apologize to people if you were having problems seeing the slides. I think there might have been a technical issue. So my apologies about that. If you have any questions, please put them in the chat box. I'm going to get the discussion going, but we'd love to address your questions if you have them. So when I was reading your book, Denis, I thought one of the things that really struck me and that it made me have a pause for thought moment was when you were talking about the Imago Dei in your last chapter, when you're talking about the Imago Dei concept and how it impacts our discussion of genetics in this day. And you said there are 4 distinct ways. Well, one of them, and you mentioned them here, was the celebration of diversity in our community. And I think that's kind of a very apt, I love that description. It's a celebration of our diversity. But it's also something that our communities, our societies, our world has been thinking very much of this past year. And so to me, that was just a comment, a thought that struck me. But it just goes to show how in our unique design that God has created us in his image, there is that genetic diversity. We're not all the same, we are unique. And that transpires or interjects into society at large as well. So I found that a very sort of, I really like that phrase of yours. So,
well, thank you. Yes, I think it is a key concept, isn't it?
yes.
Just before saying perhaps one or two things about that, let me apologize if you weren't able to see the slides. I could see them perfectly on my screen, I'm afraid, with screen sharing. So if you didn't get to see them, let me just say I'd be very happy to send the slides to anyone who wants them. Okay, and also maybe when this talk, if this talk, I think a talk is being recorded, so if it's posted on the relevant website later on, hopefully we'll get the slides together there as well. Anyway, let me just say that before saying anything else. I do think this question of diversity is incredibly, important right at the moment, isn't it? And if we just think of, well, let's think about the pandemic. We have to think about the pandemic sometime, don't we? And
Yes.
probably genetic diversity does have something to do with people's response to COVID-19. And, it comes into immunology, it comes into how easily we get infected. And so genetics gets into everything, okay? And so therefore, we need to be very concerned, I think, that we are just in our distribution of vaccines around the world, that we are very careful to make sure that low-income countries have access to really best vaccines possible, and so on. So I think there's a huge discussion there, obviously, that would probably lead us away from our main topic for today, but
Yes.
it does impinge upon many, many different aspects of our life right now. I think you're right, yeah.
Yes. We have a question from the audience that says, asking you to please elaborate when you speak of the more than 100,000 variances of our brain.
Yes, I think I might be I may have been moving too fast there without the slides. 100,000. I don't think that's a quote of something I said, but it might have been something where a little got mingled up there. I mean, clearly the brains that we have are influenced by all the genetic variants that we have. And, I mean, 50% of our genome is involved in the early development biology of our brain structure and capacity and so forth. So now, of course, our brains are very, very similar. Otherwise, we wouldn't be anatomically modern humans. We wouldn't be the humans that we are, that's for sure. But at the same time, there's tiny variations in the genes during early development and postnatal development. Does make a difference. It can make many, many differences to the structures of the brains that we have. And so the functionality of the brain will reflect its structure. And so there will be, we have people who are genius, and we have an Isaac Newton who did most of his great discoveries in his early 20s, okay, as an example, in history. Was his brain a little bit different, from my brain? I think probably it was, actually. He had some other problems. I don't know if his brain was the optimal from a social intelligence point of view, but, it was very well developed from a point of view of brilliant insights. We could say the same in Einstein and so forth. And so, I'm just saying the obvious, really, that genetic variation is very much involved in the variation that we see between us all as human individuals, but it's not deterministic. You could have a brain like Einstein, and you could choose to be a bus driver, right? So it doesn't determine what people do, but it clearly is an influence. That may not be answering that question, so you might. I'm sorry if I didn't quite understand fully the question. It's probably back to you, and maybe Jeff has some comments on that, the early field development of the brain. I'm sure that's a field that Jeff is well into, much more than I am.
Well, I'm not a developmental neurobiologist, so I work on much earlier embryos for most of what I do. But I guess, Dennis, one thing you didn't talk about in your presentation today, largely because you're trying to take a book and condense it into 30 minutes, was the second part of that, of the last chapter. So you talked about kind of the biblical view of what it means for humans to be image bearers. But then in the last chapter of your book, you juxtapose that with a discussion of transhumanism. And I found that to be quite fascinating, because it really gets at a crucial issue. What it means to be human is a crucial question relative to what does it mean to repair defects? What does it mean to enhance? And is it a good idea to enhance? And do we have the wisdom to be able to do that at all? You know, Paul Ramsey was a bioethicist. Now, he was writing in 1970, so I apologize for the gender non-neutrality of what he says here. He says this: men ought not to play God before they learn to be men. And after they've learned to be men, they will not play God. And that's a very quotable statement, I suppose, but it does presuppose what our understanding of human beings is, what their role in the world is, and what their role is before God. And most of your book kind of makes the case, any trait that you want to tweak, it's going to be really hard to do that because of multigenic traits and pleiotropy, you know, which is the idea that a single gene has many effects and all of that kind of thing. You do a wonderful job of that in the book, but I just wonder, what role do you think Christians have in discussions like this about what it means to be human? And what difference does the Christian perspective make on the way that we go about these discussions after you kind of clear away the woolly-headed nonsense, which you do a wonderful job of that?
Yeah, I think that's so important. And I think the reason I wanted to write that final chapter in particular was just to say that at the end of the day, we can discuss, you know, the differences that genes make and so on. But when you look at people's lives and what they do with their lives and how they think about new scientific discoveries, really the most powerful influence at all is this sort of top-down causation of their worldviews, as I want to call it. And so that's why I took a Christian worldview with a central idea of the image of God that we've been talking about, and wanted to contrast that with the kind of worldview that you find in transhumanism. Now, to be fair to transhumanism, you have to break it down. There are many, many strands within transhumanism, so I don't want to misrepresent any transhumanists who might be listening to this. But I think one of the central planks, beliefs in the transhumanist worldview is that we should seek, as much as we possibly can, to physically transform humans, to make them not so limited to their evolutionary histories, not so limited by their physical bodies and so forth. So we should do all that we can genetically, or in terms of AI, in terms of robotic interventions, whatever it might be, to really amplify the individual such that they become something quite different, actually, transhuman. That's what the title of the whole movement is. So, and of course, for certain transhumanists, then they think of actually downloading their minds into sort of digital space so they will live forever. And this is, you know, at the one extreme of the transhumanist movement. But I think what's so important here is to see how worldviews can really make a big difference to what we do with our science and how we utilize our science. So if we come now to the Christian worldview, well, the first thing we learn from the Bible is that God is interested in the poor. God cares for the poor. And you see that all over the prophets in the Old Testament. You see that in the New Testament. You see that in the early church. God is concerned for the poor. That's a priority for God, the underprivileged. And the problem is, of course, is that our technologies and our new scientific breakthroughs so often just benefit those who are already well off. We're already rich. We already have so much. And it takes a very long time for these benefits to trickle down to the poor and to those in less fortunate parts of the world where we may find ourselves. So I just think that is the first sort of thing I'd highlight, really, the way that Christian worldview, our Christian life, should make a dramatic difference to how what we do with our technology and so forth. Now, there's some wonderful examples whereby, you know, that Bill and Melinda Gates, I know they're not doing it particularly out of a Christian worldview, as far as I know, but using their wealth, you know, to care for many underdeveloped and poorer parts of the world, that's great. But I think that gives an example of how Christian worldview, with humankind being made in the image of God, would be central to our thinking in this particular area. It also comes into genetic engineering as well, doesn't it? So why would, okay, so are we genetically engineering to heal, or to enhance. To enhance would be much more up the transhumanist end of the spectrum, but healing is clearly central to what Jesus told Christians to do, to get on with healing others, as he himself showed us so powerfully by his own healing ministry. So again, I think that you can see how if you start with the Christian worldview and the transhumanist worldview, they begin to divide and go their separate ways into two different roads very quickly.
Thank you. I do have a question. I think we have time for one more quick question. It says, I think you mentioned that inheritability is about variance and not about what it sounds like it should mean. Okay, sounds like a bad name for what we really care about. But what is the state of figuring out how much of my behavior is genetically inherited from my parents, non-statistically?
Non-statistically. I'm afraid you can't really do that non-statistically. That's the problem. Okay. Now you might imagine you could do it. Let's just, okay. So first of all, then just about the language. It's such a big pity that in 1942, the word heritability was picked for the particular kind of behavioral genetics that I'm talking about here. And it's used in animal genetics and breeding and all kinds of things. And so inheritance and heritability are they're related, but they're two different things. And the problem is that often, even scientists sometimes, when they're reporting their results, sometimes mingle the two meanings together, which gets very confusing. So of course, inheritance is what I inherit from my parents or from my grandparents, be it money or houses or my genes. Okay, so on average then, we of course share 50-50 of our genes from our mother and father, but at the same time, during meiosis and crossing over, there's a mingling of bits of chromosome together and so forth. So it gets a bit more complicated than you might think. But clearly, yes, we are more similar, most likely, to our parents than we are to a distant cousin or to a non-related relative. But even so, you know, you look around, I know some families with a lot of kids, and I look at their parents and I think, do those kids really come from those parents? Okay. And so if you want an argument against, you know, cloning, just look at families around you. And I have, okay, so I have a brother-in-law, perhaps I shouldn't say this, but I will, who is a priest in the Church of England. And he has two daughters, and I look at them and I think, are they really from the same parents? They are just so different, okay. So I think that's a good thing to remember, okay. So once you start mixing up the genes, you know, from our parents and the grandparents and mingling them up together, you're going to get a unique human individual. And as I said in the talk, actually, we have sufficient variation in our DNA. It's an immense amount of variation in there that you can guarantee that there is sufficient variation so that every single individual in the world who ever lived or whoever will live actually will be different. Even identical twins are actually not as identical as you might think. Epigenetics, but that takes us in another direction. Okay, so
yeah. Well, unfortunately, we've run out of time, and that leads me to thank Dr. Denis Alexander for your presentation. As I mentioned earlier, it was excellent. And thank you for the engagement with the questions. And also thank you for offering to have your slides available. That's very much appreciated, and we'll make sure that there's a link or a way for you all to get those slides. Also, I'd like to thank everybody for attending. We appreciate you being involved in this session, and we wish you a good rest of the day and enjoy the rest of the conference.